Oh, right. Some more absolutely classic histology. Skeletal muscle. That is muscle that we can choose to control. Striated muscle, it's also called. Cardiac muscle is also striated. So we're just going to look at this one muscle type. God, I remember looking at this in when I was in at university. We will talk about the myofibrils, what they will have a look at them, see what they look like. We'll look at the connective tissue if we can find some to see how it's all joined together. We'll talk about the sarcomeres, what gives that stripy appearance. But yeah, let's have a look at some skeletal muscle, shall we? I've got two slides for you and a boring story. So the slide I've got here, so for the skeletal muscle slide, we're looking at the tongue. Because out of all the slides I've got, this is the best example of skeletal muscle. Now in the tongue, you've got intrinsic muscle fibres and they're running in different directions. The tongue is one of those things people have got weird ideas about the tongue. They'll say it's the only muscle that's only connected at one end. But actually the muscle is inside the mucosa, right? So the muscle is attached at either end to the connective tissue and the mucosa of the tongue. So as the muscles contract, they change the shape of the tongue. If it gets narrower, it gets longer because it keeps the same volume. Anyway, see, told you, tongue. The reason I talk on and on about the tongue is that, again, when I was at university doing my degree, my third year BSc project, if ever you're doing a science degree, there should be a third year lab-based project that you can get your teeth into. I looked at tongues. The question was, how come frogs can send their tongues so much further out than other animals? We thought it was because of the way the connective tissue was arranged around the outside of these myofibrils and what have you. Turns out, no, they cheat. They just like fold their tongues and hide them in the mouth better. Anyway, I looked at a lot of tongues, dissected a lot of tongues from lots of different animals, looked at them down in a scanning electron microscope. They left me alone with a scanning electron microscope. To be honest, that's probably one of the reasons I've ended up here, because that grabbed me, right? So, the tongue. So, because we've got all those muscle fibres running in different directions, we get to see muscle fibres in a longitudinal section, and we get to see muscle fibres cut in a transverse section. Now, the other slide I've got is black. So I assume this is a silver stain, and this will let us see the striations even better. Okay, what do we got? And looking at the gross slide, oh yeah, it's another very pretty Masson's trichrome. So we're not doing the tongue today. We'll do the tongue another day. But that there, that's the surface of the tongue. See all those spiky bits? So cool. Those spiky bits are the filiform papillae. That's the roughness of your tongue. So that's the epithelium. The green is connective tissue. And as we go deeper in here, now we're looking at blocks of muscle. So we've got some muscle fibres running in one direction, some muscle fibres running in the other direction. So we're seeing lots of red, and that's all skeletal muscle. And we're seeing lots of green, that's the connective tissue binding it all together, and in there we'll find blood vessels and nerves and what have you. Now when we say skeletal muscle, we're talking about muscle you can choose to control. You know, I can choose to control my tongue, muscles that are attached to bones and that sort of thing. The diaphragm is an interesting example. The diaphragm is skeletal muscle. I can choose to control it, I can choose to contract it and relax it. Most of the time I don't have to, purely because there are centres in my brainstem that are moving it for me, so I don't have to think about it. But skeletal muscle is muscle under voluntary control, somatic muscle. Cardiac muscle is in the heart, and smooth muscle is autonomic muscle that you can't control, just does automatic things for you. So we're looking specifically at skeletal muscle here. Now, oh, okay. That's cool. So there's lots of muscle bundles that have been cut through transversely, and if I slide up to the top, there are lots of longitudinal muscle fibres there. Let's jump down. So skeletal muscle and cardiac muscle get lumped together as striated muscle, because if you have a good microscope and a nicely stained section and you look at it right, you can see stripes, and that's why this is such classic histology, because it's the stripes we're going to talk about. Can you see how this is muscle that's been cut longitudinally, so we're seeing the length of the muscle. It's all a little bit separated. I want to find, that's a bit heavily stained. I want to find a nice kind of homogeneous bit. There are lots of interesting things about skeletal muscle cells. Maybe this bit here. Let's jump up. So this is now my 20 times objective lens and 10 times eyepiece lenses, so 200 times magnification to my eyes. Now this still isn't showing me what I want to see. So we're seeing a lot of muscle here, and of course we're seeing lots of fibrils within the muscle. Oh, that's quite good. Right. Can you see some stripes in the middle there? I have got more magnification to do, which I'll do in a minute, but can you see some stripes or imagine some stripes there? Now, one of the things that's interesting about these cells, so the cells of skeletal muscle, now a muscle cell you might call a myocyte, they're incredibly long cells. In some of the muscles of the body, these cells can be centimetres long. You can have a muscle cell that's 10 centimetres long. And we're seeing a lot of nuclei. So the purple staining bits are the nuclei. And can you see how that cell in the middle there, that red streak, those red streaks are cells. They are the myofibrils. Can you see that the nuclei, the purple nuclei, are around the outside of the cell? They're inside the cell, they're just on the outer edge. So inside the cell we've got lots of microfibrils. So the nuclei, really, they're getting out of the way of the microfibrils that are going to actually do the contraction work. So these are very long cells. We see lots of nuclei around the outer edge of the cells. This has happened because when the muscle formed, there were lots of individual cells. And then they joined up. They lost their cell membranes in between one another. This is called a syncytium. So they lots of cells joined together, lost the cell membranes that were separating them, forming very, very, very long cells, which is why there are so many nuclei, because each nucleus has come from one of those individual cells. So we've got long cells, nuclei around the outside, formed from multiple myoblasts of the progenitor cells. And this means then you've got, so of course the purpose of the muscle cell is to change length, is to generate force. And if you attach that to bones, you can then move bones. If you put it inside the tongue and attach it to the inside of the mucosa of the tongue, you can change the shape of the tongue. So can you... Oh, look at that. Do you get a strong sense there of separate, like, lanes of red? And can you see they're separated in some places? Imagine these are in 3D. So we've got bundles of these fibrils being bundled together. But can you see there's a little bit of connective tissue between some of these cells? So the cell itself is surrounded by endomysium. Like that, it's a connective tissue layer covering the cell. And then a bundle of these cells together and hold them together with perimysium. So that's like the next layer of connective tissue. If we look at, look at nerves and blood vessels. Nerves, blood vessels, right? They're in the connective tissue. So here we're looking at myofibrils cut in a transverse section. And then can you see how these cells have been bundled together? So that's surrounded by perimysium, and then the muscle as a whole is surrounded by epimysium. And that connective tissue is really important to the strength is kind of the wrong word, is really important to the strength, the structure of the muscle. So the connective tissue holds the muscle together. The muscle needs to generate a lot of force, which is why I'm struggling with the word strength. But that connective tissue is part of keeping the muscle together, but also part of the muscle pulling on other parts of the muscle. It's part of the contraction machinery, really, right? So as the muscle cells get shorter, the connective tissue supports all of that and pulls everything together so the whole muscle gets shorter. Am I making any sense? All right, so that's a nerve in the middle. So there's loads of nerves in the connective tissue. There's loads of blood vessels in the connective tissue. Look, that's a great one. It's filled with red blood cells. So the lymphatics, the small blood vessels and the nerves are within the connective tissue. So that's one of the jobs of the connective tissue as well. Okay, let's try that one. Can you see the banding? Can you see the stripes and the striation in there? That's the striations. Now, like I say, on the other section that's been stained black, we'll see this much more easily. But I'd like to see it on. I'd like to see a good one on here somewhere. How's about that in the middle there? Now this is my 40 times objective lens. I've got 100 times, but I couldn't find any mineral oil, which we could probably have done with to get up to a higher magnification. So the banding, what we're seeing is we're seeing dark bands and we're seeing light bands, and they're alternating. The banding is being caused by the contractile filaments inside the cell overlapping or not overlapping. So you've got thick filaments and thin filaments. You've got myosin and actin. Myosin of the thick filaments, actin of the thin filaments. And for contraction to occur, these filaments walk along one another, one way or the other, and both of those take energy. We're not going to talk about the mechanics of that. But you have actin and myosin, they overlap one another, and the functional unit inside the myofibra, the contractile unit, is called the sarcomere. And where those thick and thin filaments overlap, that's where we see the banding. So the actin and myosin filaments are about 10 nanometers thick. So to show you those, I'd need an electron microscope, which would be a much longer afternoon and probably less interesting for you. But where we just have thin filaments, that's the light band. Where the thick filaments then overlap with the thin filaments, that's where we see the dark band. So the thin filament, the light band gets called the I band, and the dark band gets called the A band. And if I describe it, you might imagine it right in the middle of the light band is a very, very thin dark band, which is called the Z disc. And the Z disc is the end of each sarcomere. So you've got Z disc, the Z disc, that's the sarcomere. The thin filaments are attached to the Z disc, and when the actin and the myosin walk over each other, those two Z discs get closer together. So basically this is sliding filament theory. When the actin and myosin slide over one another, the muscle contracts and gets shorter. Okay, let's see what else we can find. So the A band is the dark band, and like I say, that's largely thick filaments where they overlap with the thin filaments. That's kind of a mostly accurate way of putting it. But yeah, so the dark and light bands are where we see thick and thin filaments overlapping each other. You see that one there? How about that in the middle there? See some striations, some stripey, some bands there. I can see most of that up there. Also in there. That's quite heavily stained. I'm going through the focal plane just a tiny bit to try and help. So they're very, very faint striations. So the nuclei there look like they're inside the cell because that's just the plane of section. They're on the outer edge of the cell. It's just the shape of the tube that we're looking at. But you do get a sense, don't you, I think, of not just the stripiness, the banding, the striations, but you can see the filaments inside the muscle there. Okay. There's some good striations in there. I always find the striations quite hard to find. That's a pretty nice one. When you see the photos in the textbook, they're lovely and obvious. But the sections I have never look that good. Also, like I say, I need to get some oil for my higher powered lenses. But you get the set. Oh, yeah, I can. Oh, that. Yeah, there's a worry in science. Don't see what you expect to see, but describe what's actually there. But to my eye, I can see the dark bands, I can see the light bands, and I can definitely see a thin dark line right in the middle of those light bands, and that is the Z disc. I'm sure my brain isn't making that up. So yeah, Z disc, thin filament, thick filament in the middle making the dark band, more thin filament, next Z disc. Cool, right. That's the best one I'm going to find on there, isn't it? So let's have a look at the silver stain. Yeah? So this is very black. You know, I assume this is a silver stain. I don't know what it is. But silver is one of the things, yeah. Anywho, so we can see some quite crinkly muscle here and some quite straight muscle here. We can see some very dark stained stuff. What we want is some myofibrils that are stained just right. Not too much, not too little, but just Goldilocks. How's that? So to my eye, that striation is pretty clear now. So I assume this is just from a skeletal muscle somewhere, not from the tongue. This is just from a bog standard skeletal muscle somewhere in the body, and we can see those striations now. This is why we call this skeletal muscle striated muscle. And if we look at the muscle in the heart, which we will do in a future week, we'll see that it's also striated. It looks similar. And over here as well, how's that? What does that look like? Oh yeah, I should get a big lump of granite or something for my desk, or slate from North Wales to stop my microscope vibrating, shouldn't I? But there we go. Those are the striations, and like I say, they're caused by the overlapping contractile filaments. And that's it. Again, like all these videos, what I want to do is just kind of have a relaxed look around. Oh, that's quite cool. Rather than just showing you photos, the moving around hopefully helps you get a bit of the shape of the tissue in your mind, right? Kind of looks like a bit of shed snake skin, that. Okay, but that's it. That's skeletal muscle. That's what it looks like down the microscope. So we've talked about the myocyte being formed from multiple cells. So we see lots of nuclei around the outer edge. We've talked about the sarcomeres and the actin and myosin filaments, and how we saw some Z bands, but how that forms the banding pattern of the dark bands and the light bands. And we've talked about the connective tissue and how that holds everything together. So I guess if we've started down this this muscle path, I must have some cardiac muscle somewhere. We will be looking at another muscle next week, I guess. Well, I hope that was useful. I hope it was interesting. I found it interesting. God, I've been doing this for donkey's years and it's still interesting. Right, see you next week.